Thank you, Deborah. And thank you, Jan and Chris and Deborah, everyone at CPTR for the invitation. I have to say it really came as a surprise, and it was a nice one. Let's see if I can make this work. Okay. So the big question of the day is, how do we combine new drug candidates and existing drugs to make combinations that hopefully will work better and faster in TB patients? And as was alluded to this morning by, I think it was Dr. Cox, if we were to make every single possible permutation out of what we have at our disposal, it would take more patients than we have in the entire South Africa to run those clinical trials. And on top of that, we probably want to come up with distinct combinations for latent, active, MDR, XDR, and TB HIV. And in some cases, we might also want to think about intensive versus continuation phase. So how do we go about this right now? Where we test combinations in animal models, and most of the time we test them in the mouse model. And whether the mouse model is the right choice is a debate that I'm not getting into today. But no matter what the animal model might be, it's a very empirical approach, and it would be very sort of important in terms of building confidence to have a backup strategy that would help us understand why sometimes combination X fares better than combination Y, or why drug A synergizes with drug B or antagonizes drug B. And so this is what I'm going to talk about today. It's an approach that we're proposing to take a more rational path towards deciding what combinations to trial in human. And so for those of you who are not very familiar with the TB histopathology, I have a one slide about TB Histo 101. And I apologize for those of you who look at these every day, but I figured it would be important so that everybody would sort of understand what I'm going to talk about in the next 20 minutes. So if we want to keep things reasonably simple, we can say that we have 4 sort of major infection sites in TB disease. The first one being a completely cellular granuloma. Another one that usually appears a little later is what we call necrotic granuloma, which is made of cells that are mostly macrophages and lymphocytes. This is the darker purple donut-like shape. And in the middle, where you have the lighter pink color, is necrotic material, so it's acellular. And when these grow and they meet an airway, they cause tissue damage, they fuse with this airway, and they create what we call a cavity. And the cavity is sort of a necrotic granuloma that has fused with an airway, and here, because of the way the tissue sections are prepared, the caseous material, the necrotic material, has been washed away. But most often there is still something here that looks like cheese. That's why it's called caseum. And finally, there is the apparently uninvolved lung parenchyma, which is a compartment that we often forget, but where it is more and more evident that there are a lot of bacilli. And so these very different infection sites have 2 major implications. The first one is that the bacterial populations that reside at those different sites are quite different in terms of their metabolic activity, their microenvironment, and therefore their respective susceptibility to drugs. So not all bacterial populations are equally susceptible to drugs. That's very important. It's not the focus of my talk, but it's something that we have to keep in mind. And the second major implication is that the drug distribution in these different infection sites is not the same. They don't diffuse evenly throughout all of those different types of lesions. So we have the first implication, which is what we call pharmacodynamics, or PD. And the second implication, which we can summarize as pharmacokinetics, or PK. And what we usually refer to as PK-PD is the relationship between drug concentrations and effect, or bacterial kill. So what we want to propose is to move away from plasma-based PK-PD analysis to lesion-centric studies. And that's what I'm mostly going to talk about today. So how do we do this practically? So if this is a sort of a standard necrotic granuloma with the caseum here in the center, and the cellular layers here are mostly macrophages, and inside the box, the rest of it is uninvolved lung. What we want to do is to quantify drug concentrations in all of these different compartments. That's the first step. And the second step is to study their activity in these different compartments so that we can relate drug concentrations to activity. And this will help us answer three major questions that human studies will never be able to help us understand. The first question is, where are the bacilli that survive drug treatment for each drug in each different type of lesion? How and where does drug resistance emerge? And I'm talking about genetic drug resistance. And then if we relate this result to relative drug concentrations in all these compartments, we should be able to answer the question of whether this is likely to be due to subtherapeutic drug concentration. And the next question is, it's a big question that we just spent 2 hours discussing in the next room over there, is which animal model would be best suited to answer all these questions? And I used to believe a long time ago that this was of paramount importance to pick the right animal model, and I've completely changed my mind. I think that now, as long as we're working with animal models that develop some form of lesions that are also found in humans, the more animal models we use, the more different animal models we use, the better off we will be. And that it's very important to benchmark all of them and to recognize their respective strengths and weaknesses, and to know when we interpret the data what they are good for and what they are not so well suited to do for. So if you have an interest in the respective characteristic of each animal model, we've published a little atlas of comparative tuberculosis pathology. We don't make any profit. The profits are donated. So if you purchase it, you do two good things: you educate yourself and you donate a little bit of money to a TB hospital in rural Indonesia. And so let me now start talking about results. So having told you that all animal models are good for something, there's nothing better than Homo sapiens. And so that's where we started from. We worked together with the group of Cliff at the NIH, and also with several hospitals in South Korea. And we designed a clinical research study where we take advantage of patients who have not been responding to TB therapy for long enough that they have been given the option to go for resection, to go for lung resection, particularly when only one lung is affected. And so we have a study design whereby we can give those patients 5 study drugs. They're listed here in the yellow boxes. So there are the 3 major first-line drugs, and then kanamycin as a representative of the injectables, and moxifloxacin as a representative of the fluoroquinolones. So these patients, 3 subjects per group, receive all 5 drugs at defined time points prior to scheduled surgery. Those time points are here. So the blue arrow indicates when they receive the drugs, and the red arrows indicate where we sample plasma. And finally, at the end of the study, at the time of surgery, the piece of lung that is resected is handed over to a pathologist. The pathologist dissects out every single lesion, and those lesions are then analyzed 2 ways. Either they are homogenized, weight homogenized, and used for standard mass spectrometry analysis where we quantify drug concentrations in an absolute manner, or they are dedicated for drug imaging studies. And you will see in the next couple of slides what is meant by drug imaging using MALDI mass spectrometry. The other thing that I also wanted to mention is that in addition to this, we sample sputum at any point between drug administration and surgery, whenever the patient is able to produce it. And this is in the hope that we're going to be able to use sputum as a surrogate for specific lesion compartments in terms of drug concentrations. Okay, so we're I'm going to say 2/3 of the way through enrollment with this study, so I'm not in a position where I can show you a complete data set. And so instead, what I'm going to show you is something that we've done following a very similar study design with rabbits, again together with a group of Cliff Berry and Laura Vaya at the NIH. And so I'm really skipping, this is a lot of results combined into one slide in the interest of time. So I'm going to explain it as clearly as I can, and if something is not clear, just wave your hands and ask questions. So what we've done here is that we've measured drug concentrations of these 4 study drugs. So they are the same except for kanamycin, moxifloxacin, pyrazinamide, isoniazid, and rifampicin. And the reason we always have moxifloxacin here is we recognize that it's not a drug that's approved for tuberculosis, but not FDA approved for tuberculosis, but it's proven to be quite efficacious and useful, particularly in second-line regimen. So we are very interested in understanding why that is. So we measure concentrations of these 4 drugs in plasma versus uninvolved lung tissue versus lesion material in groups of rabbits that are again sacrificed at different times relative to drug administration. And we compare the exposure over time. So every bar here represents the ratio between exposure in lung or exposure in lesion relative to plasma for each of these drugs. And so the horizontal line here is the line of identity. Anything that's above it means favorable partitioning into either lung or lesion, and anything that's below it means that, oops, it's dead, exposure over time in lung or in lesion material is inferior to what we see in plasma. And so the striking conclusion here is that moxifloxacin partitions very favorably into lesions, while pyrazinamide and isoniazid do not seem to penetrate well into lesion material, and rifampicin appears to penetrate well into uninvolved lung, but not so well into lesions. But what we can see, and is highlighted by the red circles, is that we have a lot of variability. And so one possible reasons why we have this variability, we thought at the time, is that we were putting together in one basket all kinds of different lesions that combined cellular granulomas, coalescing, necrotic, mineralizing. So we thought this might be the reason why we see this variability, is that we don't have a good resolution, so we have to look at the lesion level. And this is what led us to move away from standard mass spectrometry looking for drug concentrations in lesion homogenates to actually imaging of these drug molecules within tissue sections. And so the way MALDI mass spectrometry works is that the whole lesion together with the surrounding lung tissue is taken out and sectioned exactly the way you would do it for histology studies. These sections are then put onto special metal plates, and they are coated with a matrix. The matrix is simply a chemical that serves the purpose of facilitating ionization of the small molecules, which is absolutely required to detect them by mass. And so the MALDI, the laser, is then fired onto the section, pixel by pixel, and it desorbs the small molecules. The small molecules then fly onto the detector, and by tuning the detector onto the mass of the molecule that is of interest, we can generate a two-dimensional image of the distribution of the drug into the lesion. And so you can see immediately the power of this system, whereby we can see the relative distribution across a section. It's also very powerful because there's no requirement for labeling the drug, unlike radioactive systems. And finally, if we know the mass of the metabolites of the drug of interest, we can also very easily look for these metabolites. And I've taken from the literature an example that illustrates very well what this technology can do, even though this has absolutely nothing to do with tuberculosis, and it's not even about drugs. But I thought it was a very nice example of what this technology can accomplish and of where we are going with this in the TB field. So, sorry, I've lost my laser pointer, but you can see here that they have looked for the distribution of 3 peptides, of which, thank you so much. this is going to be much easier. So they've looked for the distribution of 3 peptides. So the masses are indicated up here in brain tissue. And this peptide A, which is in the blue channel, distributes preferentially, as you can see, and so forth with the other 2 peptides. And so by overlaying the 3 images on top of each other, they obtain this beautiful image, which indicates that the 3 peptides actually cover the entire tissue. And in turn, when they overlaid this on top of the histology section stained by H&E, then they can sort of co-register the distribution of the peptides on top of the tissue. And so this is what we've been doing with lesions in TB-infected rabbits and in patients. So in the next slide, what I'm going to show you is the distribution of moxifloxacin again. So we were particularly puzzled about moxi because we saw this very favorable distribution using standard mass spectrometry. And so we wondered, how is this going to translate in terms of imaging? And so what we've done here is actually what I described to you with the human study. So the rabbits receive the drugs at defined time points prior to sacrifice. And at the time of sacrifice, we freeze the lesion tissue, and those are sectioned and then looked at under MALDI mass spectrometry. And so what you see here are heat maps. So red means lots of drug, and dark blue means no drug. And green is everything in between. So if you look again at this lower panel here, you can detect, circled in red and white, 2 very nice necrotic granulomas, which are made of cells on the outside layers and of necrotic material inside where you see the pink dot. And so what you see with moxifloxacin is a very favorable accumulation in the cellular layers, but a very poor ability to diffuse in an efficient manner inside a necrotic center. And so this is information that we wouldn't have never have obtained using standard mass spectrometry. And it's very important because we think that caesium is the place where a lot of slow replicating but probably very relevant bacteria reside. And so this is a compartment where we do want to have good drug distribution. So next we looked at something very similar, but this time in our human research study in South Korea. And what we see confirmed nicely what had been observed in the rabbit. So here I don't have the H&E staining, but the optical photo is clear enough because those nodules were very large to show you that the necrotic section, the necrotic center, which is outlined by the white line here, is exactly the region where we have poor distribution of moxifloxacin, while, same as in the rabbit, the cellular region here is well served by the moxifloxacin distribution. And the same is seen in those 2 other nodules that came from the same subject. This subject was dosed with moxifloxacin 4 hours prior to surgery. There's one technical point that I want to highlight, which is something important, as you'll see in the next couple of slides, is that the nature of the underlying tissue can affect the efficiency of ionization and of desorption by the laser beam. And so this needs to be corrected for. And the way we correct for it is by spraying the section very evenly with an internal standard. And by imaging this internal standard in the same section or in just the next section, either above or just under, we can subtract the image that we obtain from the raw image that we have obtained with moxifloxacin, and we then come up with a corrected or normalized image of moxifloxacin using the internal standard. All right, so next we moved on and we did something similar with this new drug candidate, PA824, the nitroimidazole. And again, here, this comes from rabbit imaging. I'm getting confused with my hands here. A rabbit piece of tissue, which again has a relatively large necrotic granuloma. The necrotic material is this light gray region here. And then here, indicated by the red arrow, you have again the region that's rich in cells, mostly macrophages. And then this lower portion here is less involved lung. I don't want to say uninvolved lung because it's quite diseased lung, but it's not clearly a granuloma material. And so interestingly, the same thing appears here. So there's relatively poor distribution of P824 in the necrotic center of this large granuloma. There's very good distribution in the cell-rich region and also very good distribution in the less involved lung region. And in this particular case, I can show you that these results were nicely corroborated using standard mass spectrometry in homogenates. We took advantage of this rabbit, which had developed lots of very nice, large, easy to work with cavities. And so we were able to, well, Laura and her team were able to separate the caesium of the cavity from the wall of the cavity, as well as the less involved lung and the plasma. And so we quantified P824 in these 4 different, let's say, compartments. And what you see nicely mirrors what we also observed using imaging. That relative to plasma, there's slightly less in uninvolved lung. There's quite a bit in the cavity caesium and much less material, much less drug in the cavity caesium. But I said, so a lot in the cavity wall, I'm sorry, and a lot less in the cavity caesium, which is in agreement with the images that we saw. Now, there's a bit of a leap here because I'm sort of treating cavity caesium as necrotic material, and this might not be the exact 2 same compartments, but we think that they are similar enough. Okay, so at this point, the big question is there anything that penetrates nicely into caesium, right? And I knew that Richard was going to be here, so I couldn't resist. I couldn't resist to squeeze the slide in. But yes, and this really got us really excited. We obtained this brand new pyrazinamide image relatively recently, and as you can see here, it was generated by a completely different type of instrument for reasons that are long to explain, but pyrazinamide is extremely challenging. And so the image might look a little bit different, but this here outlined by the yellow line is the caesium, or the necrotic center of the granuloma, and this here is the cellular region. And what you see is a relative, favorable distribution of pyrazinamide in the caesium versus the cellular region. And so this is really interesting, actually, because if you think about it, everything we know about pyrazinamide from the literature in terms of its activity in vitro, its activity in the mouse and the guinea pig, and in the hollow fiber systems, we think that it is preferentially active at slightly acidic pH, that it is preferentially active against slow replicating bacilli, and that it's for sure as active against extracellular bacilli than intracellular. And so this is sort of a good example of serendipity at work, whereby 60 years ago, when people had no concept whatsoever of PKPD because it didn't exist, they came up with a drug which favorably distributes, or appears to favorably distribute, into that particular compartment where its preferred target population resides. And this is potentially why we keep seeing that pyrazinamide is such an important component in every single drug regimen that we've tried so far. So what have I shown you so far? That pyrazinamide seems to penetrate very nicely into caesium, that moxifloxacin and P824 distribute and accumulate well into the cellular regions of granulomas, and that P824 also covers very nicely the less involved lung tissue. And so even though this needs to be confirmed and we have to get into understanding the respective activities of these 3 drugs in these different lesion compartments, it's tempting to look at this complementary distribution in light of what was recently published by the Global Alliance, which was the result of 6 combinations that were trialed in extended EBA studies, where the combination of these 3 drugs happened to perform actually better than the 4-drug combination. And so this is going to generate, we think, a lot of very interesting hypotheses that we will then be able to test in order to show that our lesion-centric PKPD approach might be a nice way to sort of confirm what we see in the mouse and to move forward with combinations that have the best promise to work faster and better in human. So I have a couple of slides about what we want to go next with this. As I have told you, this is just PK. So it's key to see whether this has anything to do with activity and how activity in lesion compartments relates to drug concentrations. And this is definitely the next step that we want to take. And the one thing that I wanted to talk about very briefly is that we really have, I think, to move beyond just counting colony-forming units on solid agar as a readout in efficacy studies. Because if you think about it, we invest so much time in animals and resources for months at a time into animal efficacy studies. And at the end of the day, we only count CFUs on solid agar, and we know very well that this doesn't give us the full picture. And so I think that there are lots of emerging tools and markers and readouts, such as fluorescent reporters, other than fluorescent reporters, how do you call it? The most probable number in liquid culture, the resuscitation promoting factors, the other growth stimulation factors. And I think all of those should be really optimized and tested and validated in order to get a fuller picture of what's happening at the lesion level in different animal models, as I pointed out earlier. And so the way we look at this in the near future is that we'll be able to work with pharmacometricians and modeling experts, and then we'll be able to compile for each drug in each type of lesion both pharmacokinetic parameters and pharmacodynamic parameters, and that somehow somebody real smart will build a software that will allow us in the future to plug in the new parameters of new drug candidates, and then the software will just magically spit out the top 3 combinations or something like that that will be then ready to test in people. Finally, one thing that our lab is particularly interested in is to sort of understand the mechanisms behind drug distribution into lesions. And the way we want to go about this is to focus on maybe only 1 or 2 drug classes and use as many members of a given drug class to understand the contribution of binding to plasma proteins and binding to tissue macromolecules in drug distribution, the contribution of active uptake and active efflux from particularly in and out of macrophages, and the physicochemical parameters that drive diffusion through caesium. And we think that by going at this in a systematic manner with 1 drug class at a time and as many molecules as possible, we will be able to decipher some of these questions. But at the end of the day, what we really want to accomplish is to move away from animals, because you can imagine that if we tell chemists in hit-to-lead or lead optimization programs that they have to make 5 grams of their molecule in order to test their ability to penetrate into rabbit lesions, this is not going to fly. So the idea that the ultimate objective is to build a couple and develop and optimize maybe 2 to 3 predictive in vitro assays that will help us understand what the tissue binding of any given molecule is inside caesium, because at the end of the day, it's the free fraction that matters, and we want to know how much free is present in the caesium. We would like to have another in vitro assay that will allow us to predict the distribution and diffusion into caesium, and finally the penetration into granuloma layers. So I'm going to skip over the next 2 slides relatively rapidly because I think they're a bit too technical, also in the interest of time, but I'm happy to discuss them. These are ideas that we have on how to come up with these new assays that we can then give to medicinal chemists, and hopefully we'll be able to engineer granuloma permeability very early on in drug discovery programs. So with this, I want to thank first off the Gates Foundation, because most of what I've shown you today was funded by them, and what was not funded by the Gates Foundation was funded by the lab of Cliff at NIH. All the rabbit work that I've shown was done in the lab of Laura, and there's a lot of people involved in the human study in Korea, very few of which are listed on this slide. And finally, the drug quantification in human and rabbit samples, the imaging and the modeling and simulation was done by us or by colleagues at Novartis when I was still there until a few months ago. I thank you for your attention.